Okay, so I'm very happy to introduce Behrouz and Andy Lee. They are co-founders of Vincere Biosciences. Vincere Biosciences conquers age-related decline through discovery and development of small molecule medicines empowered by dual platforms of AI and mitochondrial biology. Dr. Behrouz is a translational neuroscientist dedicated to the development of therapeutics for age-related diseases. Before founding Vincere, she worked with Matt Farrer at Mayo Clinic to characterize genetic models of Parkinson's disease, and was part of the team that discovered a new pathogenic mutation in VPS35. Andy Lee brings a lifetime of software and systems engineering, previously VP of engineering for a Fortune 100 banking platform serving 90% of its US market. He is co-inventor and lead developer of the stack of patented AI tools powering Vincere's drug discovery. Welcome.
Well, does this work also? Yes. Okay, great. Thank you so much, and thank you for inviting us. It's great to be here from Boston. It's starting to get a little cold up there, so we're always happy to escape when it's possible. Quick question before we get started. How many people here would be amenable to working with their spouse? That's a good number. How many people would absolutely not want to do it? Okay, it's about half and half. This is a good audience. So I lead with this because we're a husband and wife team. You'll see throughout the presentation that our paths have been quite different, and we're just going to kind of give you the story and walk you through how we started and how we ended up actually being interested in aging, because neither of us have come from the aging field itself. So my background is Parkinson's disease. I've been interested in neurodegenerative diseases. They're age-related. There's nothing that modifies these diseases. And of course, we need to really develop something. And the path has been a relatively straightforward one. So we formed the company around Parkinson's disease to develop disease-modifying therapeutics for this disease. We landed on mitochondria as a very important player in the disease. And then, of course, that led us to aging, because aging, mitochondria are one of the hallmarks, some of the contributors to aging. And Andy's background is completely different, but you'll see that it intertwines quite heavily here.
So my background is in computation. I wrote my first lines of code in elementary school, and then have been doing it professionally for a couple of decades through a few industries before joining Spring in the fight for a cure for Parkinson's disease. And it has been thrilling to learn this space and move things forward. So across these areas, I've really come up with a recipe for pushing the limits of complex systems. And this works whether you're trying to modify a car or overhaul a trillion-dollar industry, and it's been quite effective. And it's really 3 simple steps of gain a detailed mechanistic understanding of the system, identify bottlenecks of that system, and then make exponential improvements in those bottlenecks. And this has served well in my last pre-entrepreneurship role as VP of engineering, where I got kind of thrown into the fire in 2008 in one of the largest mortgage servicing companies in the country, and had to massively overhaul a system that overnight had orders of magnitude more volume than they were designed to handle, bottlenecked on one side by technology from the 1960s, and on the other side with regulatory changes that were shifting under our feet every day. And so to do that, we had to put this in process, and very quickly captured 90% of the US market with the platform that we had. So once that subsided and I was able to catch my breath, we had an opportunity to start to look in the evenings and weekends and start to talk about, with Spring about, can we apply this approach to Parkinson's disease? And 2 real questions. Can we get enough of a mechanistic understanding of the disease process to make a plan? And then what are the main bottlenecks to discovering an effective drug for the disease?
Right, so for Parkinson's disease, as I mentioned, I worked on the genetics side of things, and that was because in Parkinson's disease, by the time that you see the patients and you see their brain, so much has happened. You have a lot of correlations. You have neurodegeneration, you have inflammation, you have these protein Lewy bodies. So it's really kind of hard to tell basically anything you think of is going wrong in the brain at that point. It's really hard to tell where the cause and effect relationship is. But there are about a dozen or so genetic mutations that have been discovered, and I was very lucky to be in the lab of one of the most renowned geneticists at Mayo Clinic. And so these give us that nice cause and effect relationship. And you can see them here, with relatively high penetrance. There are some risk factors also that, with a little bit lower penetrance contribute to the disease. But we wanted to just look at the high penetrance ones. But just this kind of understanding didn't really help us that much because the main bottleneck was what should we drug? And if you're familiar with the Parkinson's space, some people just directly go after those genetic links. But that doesn't necessarily mean that you're going to affect the majority of the population that doesn't have those genetic links. So we wanted to ask this question of, what can we actually do? And if you look at, let's say, for example, each of the genetic links being in blue here, each of them have a ton of interactors inside the cell and contribute to a variety of different pathways. And so we have just 4 of them up here for simplicity. Imagine 12, each with their own links. So the question we wanted to ask is, what is it that they actually have in common? And this is where the computational work came in.
So building a knowledge graph of the interactome and other disease gene links and drugs, we can start to build an understanding of the system's biology within this, and then use some algorithms to rank order the degree of influence of different players within this space, and look for the level of overlap amongst that point of in the center. And what we find for Parkinson's disease is that mitochondrial quality control and the mitophagy mechanisms rise very highly to the most overrepresented within that genetic context. And even beyond the core Parkin-mediated mitophagy pathway there, you see a lot of other related things, including apoptotic process and electron transport chain and other areas within the mitochondrial space. So then the next question is, do we have enough of a mechanistic understanding of mitochondrial quality control to drill in deeper and modulate that pathway?
And it turns out we do. So the pathway around mitophagy was actually elucidated in a lot of detail lately. Richard Ewell of NIH just won the Breakthrough Prize for this last year. And I hope that you'll see the benefit of this system as one that we actually understand the players that are involved and which ones might be good players to actually target. So I'm going to give you a little quick overview of mitochondria because I think it is such an important player in aging and I think an underrepresented player. I've attended a few of these meetings now. Thank you, by the way, for accepting us that are coming from outside fields, but there hasn't been a lot of mitochondrial conversations. So I'm really happy to be here and talk about that. So if you have this mitochondria, you have a lot of, besides the oxidative phosphorylation, people know the mitochondria as the powerhouses of the cell, and then maybe you know a little bit more that they produce reactive oxygen species that can damage the cells, but there's a lot of quality control that happens with these mitochondria. So on the outer mitochondrial membrane, for example, you have proteins like MIRO, which is this little guy over here, and that tethers it to cytoskeleton, which helps it move across the cell, especially for neurons that have long axons. They have to be transported back and forth. You then have other proteins like MFN2 that tether it to either other mitochondria where they can fuse together or tether it to lysosomes and other organelles. And so there's a lot of this kind of quality control that happens with mitochondria. And this is primarily controlled through ubiquitination. So you have these E3 ligases that are on the membrane that put these ubiquitin chains on, and that modifies whether the mitochondria move or are able to fuse with other mitochondria. And then on the other side of that, you have this protein called USP30, which is a deubiquitinating enzyme. It takes these ubiquitin chains off. Remember USP30 because we're going to be talking about it quite a bit coming up. So when the mitochondria become damaged, what happens? There's a protein called PINK1. It's a kinase. It starts to accumulate on the outer mitochondrial membrane, and that starts to phosphorylate these ubiquitin chains that are on the outer mitochondrial protein. And then that recruits another protein called Parkin. And Parkin continues to then, Parkin is an E3 ligase, it continues to add a whole bunch of these ubiquitin chains on the mitochondrial membrane, and that really is the thing that starts to recruit the autophagic machinery, then engulfs the entire mitochondria into an autophagosome, and sends it to the lysosome for degradation. So we understand this pathway quite well. And the reason I say, remember, USP30 is the USP30 still the ubiquitinating enzyme. So Parkin puts these tags on, USP30 takes them off. They're kind of in constant yin and yang of each other battle to whether to clear this damaged mitochondria or not. There's a little bit of a additional pathway as well that I won't get into the detail of, but it's important because it uses the exact same machinery. And basically what it does is takes a chunk out of the mitochondria in these mitochondrial-derived vesicles and just sends that for degradation instead of breaking down the whole mitochondria. So while you see these pictures of kind of these static mitochondria, it's not like that, right? There's continuous things happening with it. They're fissioning, they're fusing, they're moving around, and they're getting broken down, with each mitochondria sticking around for 30, 40 days on average.
So zooming back out a minute, we know that dysfunctional mitochondria are causative for a variety of diseases, aging itself, loss of energy that goes along with that, and kind of a cascading problem. So we set out to see if we could make small molecules that can increase mitophagy, and through that, return the cells back to health.
And so this is one slide, but it took us quite a bit of time to get to actual small molecules that can safely increase mitophagy. If you look at the red line, you can see this is in human neural cells in a petri dish. So on the y-axis, you can see mitophagy. On the x-axis, increasing concentration of one of our molecules. And in the red line, you can see that we're able to increase mitophagy a little bit when we don't damage the mitochondria, because it shifts the threshold of, when does the cell consider this mitochondria damaged enough to bother removing it. But if you damage the mitochondria with something like oligomycin antimycin, which then is more like a disease state or an age state, the cells naturally try to remove these damaged mitochondria. And if you give the compound at that point, you're able to really robustly increase this process of mitophagy and actually clear these damaged mitochondria quite a bit more robustly. So these are, I'm a neuroscientist, so I always focus on the brain first, but we have looked at a whole variety of tissues, and what you see is it's actually really different in different tissues. So they don't all respond in the same way, particularly with how much basal mitophagy there is. I was interested the Calico presentation earlier showing that it is one of the declining factors. I think really we want to be looking at which cells are most likely to benefit from this, and they showed also that neurons are one of the ones that are more likely to benefit. ARPE cells, for example, retinal cells are another ones where we see a high level of basal mitophagy as well. I am sad to say we have not looked at ovarian cells. Did not occur to me at all. So thank you for that conversation earlier. We'll definitely be looking at that also.
So the next question is, can we increase mitophagy in vivo? And for that, we've recently licensed a really cool technology from the NIH with the MT-Kaima mice. These have a fluorescent reporter in the mitochondrial matrix that fluoresces green, and then when it goes into the acidic lysosome, it changes wavelength to red. And so with this, we're able to visualize the mitochondrial morphology, and then also the ratio of mitochondria that are undergoing mitophagy at any given time in an in vivo system. Using this, we have been able to show that our compounds do indeed increase mitophagy. This is in the hippocampus of a mouse after 5 days of dosing. And you see, again, a fairly subtle increase in basal mitophagy. There was no other disease state with this. This is just looking at the increase of basal mitophagy.
All right, so now for this crowd. We know that aging is the biggest risk factor for Parkinson's disease. Of course, that aging is the biggest risk factor for a lot of diseases. Is it actually having a causative effect here? So we know, for example, that a lot of the factors that are involved in mitochondrial function, whether it's oxidative phosphorylation or mitochondrial biogenesis or mitophagy, which is the thing we're interested in, everything declines with age. And again, that was highlighted by the Calico group earlier. It's been shown pretty consistently that it is one of the things that just declines as you age. And then, of course, people have shown that if you increase levels of these proteins that are involved in this mitophagy process, here I'm showing you Parkin, it's also been done with PINK1, you're able to increase lifespan and health span. The females are fertile longer, median and maximal lifespan are affected, and even if you do it in adulthood, you're able to enhance lifespan a little bit.
So with USP30 as well, and moving into mammalian systems, there's some emerging evidence that USP30 may also affect the aging processes, which is not terribly surprising given the mechanism. But last year, a group published that that in an aging model of the heart, using a substance DGAL, which causes accelerated aging, you get increased levels of cellular senescence. And if you overexpress Parkin or knock down USB30, so either of those mechanisms, you're able to reduce the cellular senescence in the heart. So we actually just got a grant from the National Institute on Aging to test our compounds for pharmacological effect in a similar model. Of course, models of aging are not aging, and there's all kinds of caveats that go along with this, but this allows us to, in a couple of months, get a viewpoint on the senolytic effect of these compounds. And then, of course, we're interested in looking in natural aging and other things in parallel.
And then another area that is heavily affected are kidneys. So if you damage the kidneys, you end up with a lot of mitochondrial dysfunction, and that mitochondrial dysfunction actually contributes to further kidney injury that kind of pushes them past the point of no return, where some acute kidney injury might turn into chronic forms of kidney injury. So that was another area we wanted to look at and see if our compound can have some effect. This is a rhabdomyolysis model of kidney injury. At the top, you can see serum markers of kidney injury, bun creatinine. Looking at the actual kidneys, you can also see some expression differences and some pathology. One additional nice thing that we got out of this is that our compound alone doesn't cause any kidney injury by itself, which is always nice to see. If you do cause kidney injury, our compound is able to reduce this kidney injury quite dramatically. So we're very happy to see that as well. As a neuroscientist, I always wanted to go brain first, but I think looking at some of these more acute indications may be a quicker way into the clinic where we can actually see something in human. First of all, show that it's safe. Second of all, show that there is target engagement and some efficacy in humans, which may actually accelerate the process for Parkinson's disease development as well, kind of thinking of it as like a Trojan horse way of getting our compound in there. Although kidney injury itself is a major unmet need. So we've kind of focused on one of these bottlenecks, what targets to drug, but we've actually developed, and I say we, but the computer scientist has developed a lot of ways that we can accelerate this drug development process. Another question that, fortunately, thank you for focusing on that very early on, are biomarkers. So both biomarkers of the disease that you're looking at, in this particular case, aging, and also biomarkers of your particular mode of action. And we've seen so many drugs in the neuroscience space not do this. So at the end of the trial, when it doesn't work, you don't know if, did the compound get into the right place? Did it actually have engage the target that you wanted it to? Or did it, actually do all of those things and the hypothesis was incorrect? And that makes a huge difference because then you don't have to keep repeating the trials. So biomarkers are great, and maybe you can speak to.
Yeah, it's another place where the systematic understanding is really helpful. The same kind of techniques you use to identify what's broken can be used to identify what to measure. In this case, we're fortunate these are enzymes with ubiquitination substrates, so we can measure those directly. And then, of course, looking downstream at some of the further things like inflammation or other things that happen.
And then, of course, for disease, things like kidney injury, there are quite a few biomarkers of kidney injury. For Parkinson's disease, less so. We have, UPDRS, which is a measure of motor, and amongst other things, but it's quite subjective. And then for aging, I asked this question of somebody that was speaking at Foresight last week of, what are the things that you're planning on measuring in aging? And he said, there are plenty of things you can measure. And that's exactly the problem, is there are so many things you can measure. I think obviously the epigenetic clocks are a good one to add, but what are some other functional things? And please talk to me afterwards, talk to us afterwards if you have thoughts on which ones might be better functional markers of aging. So once you have, a target, then how do you actually modulate that target? If there isn't anything in the literature that modulates that particular target, where do you start?
And this is an area that I think is really exciting, with virtual screening becoming much more applicable. Things like AlphaFold 2 getting a lot of structures, and even some emerging large language model approaches to generating protein structures that we can use for virtual screening campaigns. The virtual screening tools themselves are getting much better and more accessible. So with this, we've been able to put together a pretty good library of over 10 million commercially available compounds as a starting point. And we successfully ran a virtual screen against Parkin. Interesting case there where it's normally auto-inhibited, but there were crystal structures of the inhibited, partially activated, and fully active forms. And we were able to overlay those and identify a binding pocket that can hold it into an active form longer. and successfully did find small molecules that modulate the activity, both activators and inhibitors, of Parkin in that screen. Now carrying that forward. And it's a good tool with these others that are kind of more unbiased. You have so many potential targets that might come out of those, and most of them have no literature around them, no known modulators. So having these other next step of how do you get started is critical.
And then the last bottleneck is which patients benefit the most from this approach. And this is something that the cancer field has done really well. They subsegment the patient population. The neuroscience field is just starting to do so. We're looking at what patients may be worse as far as mitochondrial function. Obviously, those are the types of molecules that we've developed. I don't think the field of aging has really thought about this. So I wanted to put that forward for you guys to think about as well. Is everybody... Obviously, everybody's not aging the same way. Some people age slower or faster, but are some people having more mitochondrial aging, other people having more immune aging, other people having more like DNA damage? Can you subsegment the population and get a more heterogeneous group to go after? In Parkinson's disease, we also think about other ways of segmenting, like fast progressors versus slow progressors, and we have that in aging. Some people age faster versus slower. Would some of that be more appropriate for the mode of action that we're thinking about? And the way that this is done is quite cool. I'm very excited about actually producing some of this data and publishing it.
Yeah, we now have, it's tractable to do really large data analysis and AI and machine learning approaches with tens or hundreds of terabytes or more of data and billions of features coming together. And what we've been focused on is multi-omic modeling here. And when you can pull together whole genome sequencing with transcriptomics and methylation and lipidomics and these other pieces, and bring them together and identify the pieces that change amongst those and start to build composite models, I think that can be really predictive. And so we've started in the Parkinson's disease space largely because the Michael J. Fox Foundation is phenomenal, and they have the PPMI data sets that have over 50 terabytes of patient data, longitudinal every 6 months, patient and healthy controls with blood samples and all of these data collected and curated. Still a challenge to move that data and to get it into one place and to regularize it in a place you can make models, but it is a tractable problem that we've overcome. And in that space, we are seeing now subtypes of patients that have distinct signatures combining genetics and genomics.
So obviously for, our goal is to maximize impact. We've started with these kind of AI prioritized targets. We have a few more of them in the pipeline that we'd like to do with mitochondrial dynamics. We have also Parkin activators that we didn't really get to talk about here in the interest of time. And then, looking at what indications you go after, something like, I know nobody wants to do neuro because it's almost the hardest thing, I think, and I think that's why we have so few neuro disease-modifying therapeutics. So maybe, again, we can kind of Trojan horse our way in with some peripheral indications first and show that first. With that, I'd like to thank our team. And also, we've been very fortunate to have a lot of funding from funding agencies, including NIA and INDS. And last but not at all least, Michael J. Fox Foundation. If you have interest in Parkinson's disease, I highly recommend that you work with them. Happy to take any questions. Yeah, so in kidney damage, and it seems like in the majority of kidney damage, when the kidneys do become damaged through a whole variety of different ways, whether it's chemotherapy or like ischemic reperfusion or whatever the way is, it actually causes damage to the mitochondria. And then when the mitochondria become damaged, they produce all sorts of reactive oxygen species. If they burst, they can be recognized as foreign DNA potentially, and that activates the immune response. So there are a lot of kind of downstream effects of these damaged mitochondria just hanging out there. If you think about it, almost like senescent cells that, if you clear them, it's better than having them hanging out there, even though they're kind of technically still there and alive. So that's how we think the mechanism is.
And I'll add there that the kidney tissue is highly heterogeneous, and this is, I think, a learning we're able to bring from the neurospace, where there are all these subtypes to the brain, and we know, certain cell types within the kidney are more susceptible to mitochondrial damage, and cell type specific interactomes are an important thing to think about too, because you may not have protein interactions in all of the cell types, and that kind of really drives your dynamics. But within that, there are parts of the kidney, the epithelial cells seem to be especially targeted there.
In the Michael J. Fox human data set, did you see mitophagy or mitochondrial biogenesis signals coming out strong?
Yeah, I mean, anytime you have Parkinson's disease, it is one of the hallmarks there as well, yeah. The one thing, that was a question, even though there are two genetic mutations that are directly in the mitophagy pathway that cause early onset forms of Parkinson's disease, so there's always been that link. But the question has been, is it causal in the sporadic population? We always see it at the end. There's always mitochondrial dysfunction at the end. But is it causal? And we do think there's a lot of there's a lot of analysis of the omics data and other genetic data that's more subtle that we've looked at that suggests that that's the case also.
Is that in comparison to non-Parkinsonian samples?
Yeah, non-Parkinsonian and also some other groups that we call them the SWED population, that have the motor symptoms but not degeneration, as is looked at by PET scan. And those guys, they clustered immediately, separated from this group, yeah. Thank you.